Hello friends, welcome to a new lecture today. Today, let us now learn about the next amino acid which is important, which is called glycine. Glycine is, first I will write the structure of glycine. Here we have a COOH or carboxyl group on the right side, amine group on left side, and we have two hydrogen atoms present in glycine. So if you see, it has no asymmetry. See, it has no asymmetry, right? It does not have four different functional groups. It has two hydrogen atoms. So it is not chiral, right? It does not have optical isomerism, or it does not have either structural isomerism. It does not have optical and structural isomerism, but it is the smallest amino acid which is available, and it is the simplest amino acid. It is the smallest amino acid which is present in for today. It is the simple amino acid. It is non-essential amino acid. See, this glycine is a simple amino acid which has very simple structure, which is very easy for our body to synthesize. So glycine is a non-essential amino acid which is synthesized in our body. Now coming to the synthesis of glycine. See, glycine, there is a molecule called as serine. This serine, in the presence of hydroxymethyl transferase, it forms glycine. Serine, in the presence of hydroxymethyl transferase, it forms glycine. Glycine also is produced from carbon dioxide and ammonium group combined together in the presence of the enzyme glycine synthase, and it forms glycine. Okay, this glycine is also formed from a molecule called as threonine, a protein called as threonine, in the presence of the enzyme threonine aldolase. Threonine aldolase converts threonine to glycine. Okay, this is how the amino acid glycine is formed. Right, this glycine synthase, for this we require a PLP as coenzyme. It requires tetrahydrofolate, and it also requires NAD plus as coenzymes. Right, now once we have learned about the synthesis of glycine, now how are we going to degrade the glycine? That is degradation of glycine. Now glycine, first it gets converted into serine in the presence of tetrahydrofolate or the same enzyme hydroxymethyl transferase will also break down glycine from serine. This serine, in the presence of the enzyme serine dehydratase, it forms pyruvate. Okay, this serine, in the presence of serine dehydratase, it forms pyruvate. Serine also results in formation of glyoxylate. Okay, it also forms glyoxylate. This glyoxylate now divides itself into a glycolic acid and also oxalate. Okay, this glycolic acid is utilized in our body, but this oxalate is excreted in urine. So if there is any chances of increased glycine, if for example there is increased glycine due to any reason, then there is increased glycine degradation, and this oxalate is increased in urine, resulting in formation of oxalate stones in the kidney. Right? Next, there is one more reaction where this glycine, okay, gets converted to carbon dioxide and ammonium directly. Okay, next, this is about the degradation of glycine. Now, after synthesis and degradation of glycine, we also have glycine is also useful for the production of some special products. What are the special products which are formed from glycine? So special products which are formed from glycine includes, first we have glutamate combines with cysteine. Sorry, I'll write in the next page. Right. Special products are, first we have glutamate combines with cysteine, and in the presence of gamma glutamyl cysteine synthase. Glutamate in the presence of cysteine glutamate along with cysteine in the presence of gamma glutamyl cysteine synthase, it forms gamma glutamyl cysteine. Okay, and for this reaction ATP is utilized and it forms ADP and inorganic phosphate. Glutamate combines with cysteine in the presence of gamma glutamyl cysteine synthase and it forms gamma glutamyl cysteine. Now this gamma glutamyl cysteine combines with the amino acid glycine and it forms glutathione synthase. This gamma glutamyl cysteine combines with glycine in the presence of glutathione synthase, it forms glutathione. So even if you do not remember these steps, remember the amino acids which are present in glutathione. It's simple. Glue is glutamic acid. Glue is glutamic acid. Gluta is glutamic acid. Okay. Thio is nothing but sulfur. So sulfur containing amino acid is cysteine. Okay. This is ONA. ONA is nothing but first amino acid which is glycine. So glutathione contains glutamic acid or glutamate, cysteine and glycine. So glutamate combines with cysteine in the presence of gamma glutamyl synthase, it forms gamma glutamyl cysteine. This gamma glutamyl cysteine in the presence of the enzyme glutathione synthase along with glycine, it forms glutathione. We have second special compound where, see here, arginine combines with glycine and in the presence of arginine glycine transamidase. Arginine combines with glycine in the presence of arginine glycine transamidase, it forms guanidoacetate. Okay, and during this process ornithine is released. Okay, arginine combines with glycine in the presence of arginine glycine transamidase, it forms guanidoacetate. Right, this guanidoacetate in the presence of S-adenosyl methionine, which gets converted to S-adenosyl homocysteine in the presence of the enzyme guanidoacetate methyl transferase. In the presence of the enzyme guanidoacetate methyl transferase, it forms creatine. This creatine in the presence of the enzyme creatine kinase, it forms creatine phosphate. In the presence of the enzyme creatine kinase, it forms creatine phosphate, and during this process ATP gets converted to ADP. This creatine also gets converted to creatinine, which is excreted in urine. This creatine phosphate will inorganic phosphate is removed and it can also form creatinine. So creatinine is produced from the amino acids which include arginine, glycine and methionine. See, first arginine and glycine in the presence of the enzyme arginine glycine transamidase, it forms guanidoacetate. This guanidoacetate combines with S-adenyl methionine, which and in the presence of the enzyme guanidoacetate methyl transferase, it forms creatinine and S-adenosyl homocysteine. This creatine now combines with ATP and creatine kinase being the enzyme, it forms creatine phosphate. In this reaction, ATP gets converted to ADP. This creatine phosphate removes a phosphate group and it forms creatinine. Similarly, creatine removes a water group, and forms creatinine. This creatinine is excreted in the urine. Then Then we also have one more, which is conjugation. One more special compounds are by conjugation. In conjugation, the cholic acid, cholic acid combines with glycine and it forms glycocholic acid. Cholic acid combines with glycine and it forms glycocholic acid. Whereas quinodeoxycholic acid also combine with glycine and it results in the formation of glycoquinodeoxycholic acid. Okay, and the compound benzoic acid combines with glycine and it forms hippuric acid. Benzoic acid combines with glycine and it forms hippuric acid. So these are the reactions on conjugation with glycine. Now, what are the uses of glycine? Uses are glycine with the help of glutathione. Glycine forms glutathione which will maintain the red blood cell integrity. Okay, glutathione with the help of the enzyme glutathione S transferase. This glutathione S transferase is involved in conjugation, that too phase 2 of conjugation. Right now, so this is about the glycine. So thank you guys for watching my lecture. Thank you.